everyone, we are the group 6 and today we are going to talk about genetic engineering or genetic modification. So what is genetic engineering? Genetic engineering is a completely artificial process where an organism's genetic composition is modified via recombinant DNA technology. During the genetic engineering process, the gene introduced to alter the natural genetic composition is known. The gene of interest is cloned onto a compatible vector. Next is, what is genetic modification? Genetic modification can take place in the natural environment and can happen under natural conditions. Therefore, it is completely not artificial or in vitro. During genetic modification, recombinant DNA technology molecular biological methods are not involved. Genetic engineering is a form of genetic modification. Genetic modification involves the union of gametes, and the modifications are passed from one generation to the next. Some common genetic modification techniques among plants include hybridization, selection, and include mutation. In plant breeding, crossing disease-resistant varieties with disease-susceptible varieties are over few generations will ultimately produce disease-resistant varieties. Plant breeding techniques, used since many thousand years, is a classic example of obtaining genetically modified foods. Both genetic engineering and genetic modification processes give rise to genetically altered organisms. Both genetic engineering and genetic modification processes are used to introduce positive characteristics to the host organism. So how did genetic engineering started? Genetic engineering, based on recombination, was pioneered in 1973 by American biochemists Stanley N. Cohen and Herbert W. Boyer, who were among the first to cut DNA into fragments, rejoin different fragments, and insert the new genes into E. coli bacteria, which then reproduced. Genetic engineering has been used to produce proteins derived from humans and other sources in organisms that normally cannot synthesize these proteins. Human insulin synthesizing bacteria were developed in 1979 and were first used as a treatment in 1982. In 1988, the first human antibodies were produced in plants. Since 1973 until now, genetic engineering is still being studied by scientists. So what is the purpose of genetic engineering? Genetic engineering allows genes from one organism to be inserted into a cell of a different organism of a different species. For example, are human genes to be inserted into bacterium? Human genes can be inserted into animal cells, and bacterium genes can be inserted into plant cells.
So there are three different kinds of genetic engineering. First is analytical genetic engineering. The second one is applied genetic engineering. And the third one is chemical genetic engineering. So we talk about the analytical genetic engineering. Analytical genetic engineering, this is the branch of genetic engineering study in which computer software is used to generate virtual genetic models. Various computers programs are used to explore the theoretical implications of various genetic engineering activities if they are to be implemented in actual. For example, building an analytically model based on an adequate program designed for the purpose before slicing two separate genes in practice will offer the researchers an idea whether such splicing would be successful at all, and if successful, if the desired end would be accomplished. This is a more efficient method of conducting the trials and error stage, and it lowers the danger of disaster during trials with live organisms, particularly animals. So the second one is applied genetic engineering. Applied genetic engineering, as the name suggests, is that field of genetic engineering which pertain to practical application of genetic engineering tools to manipulate the genes of living organisms for making genetic copies of them, or to introduce certain different characteristics in them that are not usually for the subjects. The first instance is what we typically refers to as cloning, and this second instance refers to the promises of transgenes. While, while cloning is a highly regulated and controversial field, it has been carried out of the various subject of animal and plant species, which makes result and uncertain success rates. Transgenes, on the other hand, is a comparatively common area, and most of us have partaken of the result of transgency sometimes or the other. So the last one is chemical genetic engineering. Chemical genetic engineering is the most basic form of applied genetic engineering, as it involves isolating, classifying, and charting genes in order to prepare them for use in applied genetic engineering activities and studies. Genetic mapping, genetic interaction, and genetic coding are all aspects of chemical genetic engineering. Following the completion of a subject DNA sequencing, DNA fragments are assigned to individual chromosomes, resulting in the creation of a genetic map. So, genetic mapping is critical for identifying the disease gene relationship, which is necessary for developing various gene therapies. Researchers can learn which genes and combinations of genes form of specific phenotype or collection of morphological, psychological, and behavior features by studying genetics interactions. Genetic coding is concerned with research and experimentation with amino acid sequence of DNA and RNA so as to understand the heredity trends and characteristic of the subject. This helps in understanding the basis, possibilities, and conditions of undesirable heredity characteristic defects and disease in a bid to come out with medical solutions for the same. So those were the three major categories of genetic engineering that have been developed till date. Other than the aforementioned genetic engineering types, two emerging fields in genetic engineering are somatic cell engineering and germline engineering. Both of these are at present and the most significant types of genetic engineering in humans, and both are used in gene therapy for correcting defective genes and for preventing the transmissions of heredity defects or disease from one generation to subsequent generations.
Genetic engineering. In medicine, genetic engineering has been used to mass produce insulin, human growth hormones, follistatin, for treating infertility, human albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many other drugs. In research, organisms are genetically engineered to discover the functions of certain genes. And this is the pros of genetic engineering: fighting disease, increase in life expectancy, increased variety of foods and drinks, nutritious foods, decrease in the use of pesticides, medical foods, decrease in the use of resources, increase in growth rates of animals and plants, development of specific characters, and this is the cons. Religious and ethic concerns, genetic issues, health issues, allergies, resistant insect and pests, antibiotic resistance, reduction of genetic variety, effect on wildlife, soil pollution, displacement of natural species, influence of certain industries and interest groups.
Now let's tackle the examples of genetic engineering. There are three examples of genetic engineering, which are plant-based genetic engineering with animals, and human and medical genetic engineering. For plant-based genetic engineering, pesticide-resistant rapeseed plants. Rapeseed is a flowering plant used to make certain types of vegetable oil. Genetic engineering has allowed these plants to be resistant to certain types of pesticides so that when the fields are treated to remove pests, the plants will remain unscathed. Plants that fight pollution. Poplar trees developed by scientists at the University of Washington can absorb polluted water through their roots and then clean it before the water is released back into the air. The plants were many times more efficient at cleaning certain pollutants than regular poplars. Golden rice. Genetic modification is often used to make healthier foods, such as golden rice, which contains beta-carotene, the very same vitamin that makes carrots orange. The result is that people without access to many vitamins will get a healthy dose of vitamin A when the rice is consumed. Faster growing trees. Demand for wood can be met by trees that grow faster than average. Genetic engineering has produced trees that can ward off biological attacks, grow more quickly and strongly, and create better wood than trees that are not genetically modified. Bigger, longer-lasting tomatoes. When tomatoes are genetically engineered, they can be made bigger and more robust. These are engineered to produce tomatoes that can remain fresh for longer, be shipped farther from where they are grown, and be harvested at the same time rather than harvesting only parts of a field at each harvest. Insecticide corn. Instead of spraying insecticide on plants, why not genetically engineer crops that kill pests on their own? Corn was developed through genetic engineering to produce a poison that kills insects. While this corn may also harm beneficial insects, such as butterflies, supporters say that the pros outweighs the cons. Next is the non-crying onions. Onions and crying seem to go hand in hand, but not anymore. In 2008, a New Zealand scientist engineered an onion without the enzymes that tears you up called sunion. These sweeter onions won't make you cry. Next,
Genetic engineering with animals. Cloning example. One of the most controversial uses of genetic engineering has been cloning, or producing a genetically identical copy of an organism. While the ethics of cloning are hotly debated, the first ever sheep, named Dolly, was cloned in 1996 by scientists. In 2020, scientists cloned the endangered black-footed parrot. Glow-in-the-dark cats. It sounds strange, but in 2007, scientists in South Korea altered the DNA of a kitty so that its pure would glow in the dark. They then cloned other cats from it, making the world's first glowing cats. Cows that pass less gas. Methane is produced by cow flatulence, and the chemical is a huge contributor to global warming. Cows that part less than average have been produced to fight the deleterious effects that how flatulence can have on the environment. Environmentally friendly pigs. Genetic modification has helped create pigs that can better digest phosphorus, decreasing the pig's phosphorus output. The result is that manure, with which is often made from pig waste, is less destructive to the environment due to its lower phosphorus content. Salmon that grows faster. Salmon do not produce growth hormones year-round, so scientists have looked toward genetic engineering and found a solution. A modification that allows salmon to grow twice as fast as those that are not engineered was the answer.
Human and medical genetic engineering. First, the banana vaccine. Bananas were developed through a genetic modification that offers a vaccine against diseases such as cholera and hepatitis. Just like a needle vaccine, people who eat them develop disease-combating antibodies that make them immune to a disease. Second is the COVID vaccine. Amid the COVID-19 pandemic, the COVID-19 vaccine used genetic engineering to achieve immunity. The Pfizer and Moderna vaccines use mRNA genetic sequencing to help a person's body recognize the COVID-19 virus. Third is the eugenics example. While not currently available, scientists are working with germline editing for reproduction. This could work to ensure children aren't born with specific defects and undesirable traits. Next is synthetic human growth hormone. Scientists have worked to understand human growth and disorders for a long time. Using genetic engineering, they were able to create a synthetic human growth hormones for individuals born with human growth hormone deficiencies. Next is genetically engineered antivenom. Rather than injecting animals with venoms and extracting the antibodies, scientists are using genetically modified DNA to replicate venoms by creating synthetic DNA. DNA that replicates the venom works to produce the immunogens in the body for the toxins. The last is the heparin example. Heparin is a medication to help with blood clotting disorders that is derived from animals. However, scientists are working to create a genetically modified version of heparin that doesn't require animal sources.
Advantage of genetic engineering. Number 1. It allows for a faster growth rate. Genetic engineering allows of plants or animals to be modified so their maturity can occur at a quicker pace. Engineering can allow this maturity to occur outside of the normal growth conditions that are favorable without genetic change as well, even if there is higher levels of heat or lower levels of light. It becomes possible to expand what can be growth in those conditions. Number 2, it can create an extended life. Genetic modification can help to create resistant to common forms of organism that resistant can be included into the genetic profiles of plants. So they can mature as a crop without any further additives. Animals can have their genetic profiles modified to reduce the risk of common health concerns that may affect the breed or species. This create the potential for an extended lifespan for each organism. Number 3, specific traits can be developed. Plants and animals can have specific traits developed through genetic engineering that can make them more attractive to use or consumptions. Different colors can be created to produce a wider range of produced. Animals can be modified to produce more milk, grow more muscle tissues, or produce of different coats so that a wider range of fabricants can be created. Number 4. New product can be created. With genetic engineering, new product can be created by adding or combining different profiles together. One of example of this is to take a specific product such as potato and alter its profile so that it can produce more nutrients per kcal. That without the genetic engineering, this make it possible for more people to get what they need nutritionally, even if their food access is limited, and this could potentially reduce global food insecurity. Number 5. Greater yields can be produced. Genetic engineering can also change the traits of plant or animals so that they produce greater yields per plant. More fruits can be produced per tree, which create a greater food supply and more profits for a farmer. It also creates the potential for using modified organisms in multiple ways. Because there is a greater yields available, modified corn, for example, can be used for specific purposes such as animal feed, ethanol, or largest crops for human consumptions. Number 6. Risk of the local water supply are reduced. Because farmers and growers do not need to apply as many pesticides or herbicides to their croplands due to genetic engineering. Firmer application to the soil need to occur. This protect the local watershed and reduce the risk of adverse even occurring without risking the yield and profitability that is needed. 7. It is a scientific practice that has been in place for millennia. Humans in the past may not have been able to directly modify the DNA of a plant or animal in laboratory, but they still practice genetic engineering through selective breeding and cross-specific or cross-breeding. People would identify the specific traits, six out of other plants or animals had similar traits, and then breed them together to create a specific result. Genetic engineering just speed of this process and can predict an outcome without greater regularity.
The disadvantages of genetic engineering. The nutritional value of foods can be less. When animals grow and mature quickly, the nutritional value of that product can be reduced. This can be seen in poultry products today with the white stripping that is found in the meat products. Number 2 are pathogens adapt to the new genetic profiles. Genetic engineering can create a natural resistance against certain pathogens for plants and animals, but the natural evolutionary process is geared toward creating pathways. Bacteria and viruses evolve a resistance to the resistance that is created by the genetic engineering efforts. This causes the pathogens to become stronger and more resistant than they normally would be, potentially creating future health concerns that are unforeseen. There can be negative side effects that are unexpected. Genetic engineering is guaranteed to make a change. Many of those changes are positive, creating more and healthier foods. Some of those changes, however, can be negative and unexpected. Making a plant become more tolerant to drought might also make the plant become less tolerant to direct sunlight. Animals may be modified to produce more milk but have a shortened lifespan. At the same time, so farmers suffer a greater livestock. 4. The amount of diversity developed can be less favorable. At some point, genetically engineered plants and animals make it into the wild and interact with domestic species. This results in a crossing of natural and artificial organisms. The engineered organisms often dominate, resulting in only a modified species over several generations, reducing the diversity that is available. Number 5. Copyrighted genetic engineering can have costly consequences. Many companies copyright their genetic engineering processes or products to maintain their profitability. If a farmer plants genetically modified crops and the pollination process causes another farmer in the field over to have those modified crops grow, there have been precedents for legal actions against the unauthorized farmer. And lastly, this knowledge and technology can be easily abused. At the moment, genetic engineering in humans is used to treat specific disorders that threaten the health or well-being of individuals. In time, the approach in humans would be like what is already being done with plants and animals. Genetic engineering can change specific traits which would create human outcomes that are ethically questionable or easily abused.